Now look at this.
What's up, Hippo Penguins? Today we're going to do topic 2.9, mechanisms of transport. Topic 2.9 has a lot of repeat information in it, so sorry y'all. So as we talked about before, there's two different types, passive transport and active transport. Passive transport is going to involve going down a concentration gradient, so it goes from high concentration to low concentration. Active transport is going to involve when we go from low concentration to high concentration. Passive transport does not require ATP. Active transport does require ATP. When we think about passive transport, there's different types. There's simple diffusion. Simple diffusion means that it goes directly through the plasma membrane. It doesn't require any type of transport protein. It can just diffuse straight through. So this would be like carbon dioxide, oxygen, steroids, like lipid steroids. Those are things that can pass directly through. The other type is facilitated diffusion or facilitated transport. This has to do with that there is a protein in the membrane that's going to allow things to move across. So for example, ions. Ions are charged particles. Because of the fact that they're charged, they're hydrophilic and they will not dissolve in the hydrophobic core of the plasma membrane. So we need to have some type of channel protein or carrier protein to move them across. Same as water. If you're moving large quantities of water across the membrane, you need some type of channel protein. There's a lot of similarities between simple diffusion and facilitated diffusion. The things that are similar between them is the fact that they're both going to go down the concentration gradient. They're both going through passive transport. There's also differences between them. A difference between facilitated and simple diffusion is facilitated has a protein while simple diffusion does not. Active transport, as we said before, is going to move things against the concentration gradient. So when we look at active transport, we're just seeing that things are going from a low concentration to a high concentration, and that we're using energy in the form of ATP or some other type of gradient. In terms of bulk transport, when we're trying to move large molecules across the membrane, we're going to go through exocytosis for things to exit. So when we think about the pathway, we're going to synthesize proteins in the rough ER. Those proteins from the rough ER are then going to go to the Golgi, and then they're going to move to the plasma membrane. And that's when we go through exocytosis to allow those proteins to be secreted from the cell. Endocytosis when we're bringing large particles into the cell. So phagocytosis is cellular eating. This is when we're going to bring large food particles into the cell. Pinocytosis is cellular drinking, where we're going to bring extracellular fluid into the cell. And receptor-mediated endocytosis is when we're going to kind of have ligands binding that cause the invagination that brings materials into the cell. Remember, I'm doing this for every single topic, so hit that follow button, and AP Bio Penguins, just success. Bye, y'all.